A syncytium or symplasm, plural syncytia, from Greek, syn. Together, plus kaitos. Box, i.e. cell, is a multinucleated cell that can result from multiple cell fusions of uninuclear cells, i.e., cells with a single nucleus, in contrast to a koinocyte, which can result from multiple nuclear divisions without accompanying cytokinesis. The term may also refer to cells interconnected by specialized membrane with gap junctions, as seen in the heart muscle cells and certain smooth muscle cells, which are synchronized electrically in an action potential. Another, correct and well-established, use of the word syncytium is found in embryogenesis to refer to the choanocytic blastoderm embryos of invertebrates, such as Drosophila melanogaster. Physiological examples. Protists. In protists, syncytia can be found in some rosarians, e.g., chlorarachniophytes, plasmodiophorids, haplosporidians, and acellular slime molds, dictyostelids, amoebozoans, and acracids, excavates. Plants. Some examples of plant syncytia, which result during plant development, include developing endosperm, the non-articulated laticifers, the plasmodial tapetum, and the nacellar plasmodium of the family Podostemaceae. Fungi. A syncytium is the normal cell structure for many fungi. Most fungi of Basidiomycota exist as a dikaryon in which thread-like cells of the mycelium are partially partitioned into segments each containing two differing nuclei called a heterokaryon. Animals. Skeletal muscle. A classic example of a syncytium is the formation of skeletal muscle. Large skeletal muscle fibers form by the fusion of thousands of individual muscle cells. The multinucleated, symplastic arrangement is important in pathologic states such as myopathy, where focal necrosis, death of a portion of a skeletal muscle fibers does not result in necrosis of the adjacent sections of that same skeletal muscle fiber, because those adjacent sections have their own nuclear material. Thus, myopathy is usually associated with such segmental necrosis, but with some of the surviving segments being functionally cut off from their nerve supply via loss of continuity with the neuromuscular junction. Cardiac muscle. The syncytium of cardiac muscle is important because it allows rapid coordinated contraction of muscles along their entire length. Action potentials propagate along the surface of the muscle fiber from the point of synaptic contact, through intercalated discs. Although a syncytium cardiac muscle differs because the cells are not long and multinucleated. Cardiac tissue is therefore described as a functional syncytium, as opposed to the true syncytium of skeletal muscle. Osteoclasts. Certain animal immune-derived cells may form aggregate cells also, for example the osteoclast cells responsible for bone resorption. Placenta. Another important vertebrate syncytium is in the placenta of placental mammals. Embryo-derived cells that form the interface with the maternal bloodstream fuse together to form a multinucleated barrier, the syncytiotropoblast. This is probably important to limit the exchange of migratory cells between the developing embryo and the body of the mother, as some blood cells are specialized to be able to insert themselves between adjacent epithelial cells. The syncytial epithelium of the placenta does not provide such an access path from the maternal circulation into the embryo. Glass sponges. Much of the body of hexactinellid sponges is composed of syncytial tissue. This allows them to form their large siliceous spicules exclusively inside their cells. Tegument, helminth. The fine structure of tegument is essentially the same in both the cestodes and trematodes. A typical tegument is 7 to 16 micrometers thick, with distinct layers. It is a syncytium consisting of multinucleated tissues with no distinct cell boundaries. The outer zone of the syncytium, called the distal cytoplasm, is lined with a plasma membrane. This plasma membrane is in turn associated with a layer of carbohydrate-containing macromolecules known as the glycocalyx, that varies in thickness from one species to another. The distal cytoplasm is connected to the inner layer called the proximal cytoplasm, which is the cellular region or cyton or pericardia, through cytoplasmic tubes that are composed of microtubules. The proximal cytoplasm contains nuclei, endoplasmic reticulum, Golgi complex, mitochondria, ribosomes, glycogen deposits, and numerous vesicles. The internal-most layer is bounded by a layer of connective tissue known as the basal lamina. The basal lamina is followed by a thick layer of muscle. Pathological examples: viral infection. Syncytia can also form when cells are infected with certain types of viruses, notably HSV-1, HIV, MEV, and pneumoviruses, e.g. respiratory syncytial virus (RSV). These syncytial formations create distinctive cytopathic effects when seen in permissive cells. Because many cells fuse together, syncytium are also known as multinucleated giant cells, or polykaryocytes. During infection, viral fusion proteins used by the virus to enter the cell are transported to the cell surface, where they can cause the host cell membrane to fuse with neighboring cells. Reoviridae. Typically, the viral families that can cause syncytium are enveloped because viral envelope proteins on the surface of the host cell are needed to fuse with other cells. But, certain members of the Reoviridae family are notable exceptions due to a unique set of proteins known as fusion-associated small transmembrane fast proteins. Reovirus-induced syncytium formation is not found in humans, but is found in a number of other species and is caused by fusogenic orthorheoviruses. These fusogenic orthorheoviruses include reptilian orthorheovirus, avian orthorheovirus, Nelson Bay orthorheovirus, and baboon orthorheovirus. HIV. HIV infects CD4+ T cells and makes them produce viral proteins, including fusion proteins. Then, the cells begin to display surface HIV glycoproteins, which are antigenic. Normally, a cytotoxic T cell will immediately come to inject lymphotoxins, such as perforin or granzyme, that will kill the infected T helper cell. However, if T helper cells are nearby, the GP41 HIV receptors displayed on the surface of the T helper cell will bind to other similar lymphocytes. This makes dozens of T helper cells fuse cell membranes into a giant, non-functional syncytium, which allows the HIV virion to kill many T helper cells by infecting only one. See also: References.